Semen volume is another parameter that determines the status of fertility or infertility. So precise measurement of volume should be taken, and the normal in case of WHO 2010 criteria, the normal semen volume should be equal to or greater than 1.5 ml, and it should be up to 4 ml, and more than 4 ml would considered as abnormally high volume of semen. In previous version 99 WHO 99 version, it was greater than 2 ml, but now it is greater than or equal to 1.5 ml. So the normal volume is required for or accurate volume is required for the calculation of total number of spermatozoa and non-sperm cells. So volume of any liquid is best measured by its calculation of its weight. So because volume is a function of mass by density, so if we divide mass of a liquid by its density, then it would be the volume of that liquid. So by using this method, we have to take a pre-weighted sample in a clean disposable container and then weigh it, and then we have already weighed the container, so we can subtract the weight of the container from the final weight of the sample and container, and then we can calculate by keeping the density of semen 1 gram per ml. So we assume it, so that would be the volume. And now the volume of the so density of the semen, it varies by 1.043 and 1.102 gram per ml. So we keep these values in this formula, that is volume is equal to mass by density. So it is around 1 gram per ml. So there would be volume. Another method of determination of volume is we can take a negative pressure pipette. So we can take, or we can use positive pressure pipette. So we can take semen sample in a pipette and can measure, or by using a syringe, and we can measure how much volume it has taken. So that would be the volume of semen. And then the why the semen volume could be abnormal, it could be low because of obstruction of the ejaculatory ducts, could be absence of the vasa deferens, or it could be the retrograde ejaculation. So semen has entered into the urinary bladder, and then it could be due to androgen deficiency because it stimulates the uh, testes to produce its fluid, stimulates all the accessory glands to produce their fluid, and then there could be inadequate erection and improper mood at collection. For example, the person that has given samples would be uncomfortable at the laboratory situations, and could not be that stimulated to release a lot of volume. And then there could be incomplete collection. We know there are 4 fractions, so the last fraction could be absent from this volume. So then high semen volume may reflect the inflammation of the accessory glands. So the accessory glands, they are producing more uh, fluids, more secretion. We know uh, maximum fractions are coming from prostate gland and seminal vesicle. So inflammation of these glands would increase the volume of semen. Semen pH is toward basic all the time, and it increases with passage of time when semen passes through the reproductive tract, because the glands that release, accessory glands that are releasing their secretions, they release basic type of secretion. So semen is the strongest buffer in the body, and its reference value is greater than 7 point, equal to or greater than 7.2. So it should be measured after 30 minutes of the liquefaction, and should be measured within 1 hour of ejaculation. So it could become more basic when carbon dioxide releases from the sample. So procedure is like we do for every liquid, that we have to use either pH meter or pH paper. So pH paper that we use should be ranging from 6 to 10, because it is always toward basic side. The high pH values are present in semen. And then if pH is less than 7, it means there is certain ejaculatory duct obstruction, or could be absence of seminal vesicles, or the seminal vesicles, they are poorly developed, so they cannot release their basic liquid into the semen. So the semen is toward little acidic. Or if semen is contaminated with urine, it is the normal function of the reproductive tract to expel the semen that is present after the ejaculation. So if that semen, that urine is contaminating the sample, then semen pH would become low, it would become acidic. So the basic type of, if the semen pH is more basic, it is highly basic, then it would give a little information, because it is normal function of the semen to become basic after a certain time period, because carbon dioxide releases out of the semen and it becomes more basic. In order to determine the other parameters of semen, we need to mix the liquefied semen sample well, and then we have to take a small portion of that sample. We have to place it on a standard volume on the clean glass slide, and then cover it with a cover slip. The depth of the glass slide or chamber, that like we are using in hemocytometer, it should be 20 micrometer deep. And then we have to keep the sample at rest, and depth of the chamber in which we are observing should be up to 20 micrometer. If it is less than 20 micrometer, it would hinder the rotational movement, and we would have a increased number of asthenospermia, that is decrease in motility, and that would be due to chamber actually. So if chamber is too deep, then we cannot assess the spermatozoa while moving. So we have to keep it up to 20 micrometer. So if the number of spermatozoa per visual field, that is the under vision, the area that is under vision, they are more, it or they would be less, it means the sample is not homogeneous. So the sample should be uh, homogenized first in order to observe it under microscope. So there could be other causes of lacking homogeneity, and these could be abnormal consistency. The contents in the semen, they are not in normal form, so there would be a lot of portion of semen would be in fluid form, and the could be due to abnormal liquefaction, and could be aggregation of spermatozoa in a certain region due to antibodies, could be due to sperm agglutination, so it could be due to inflammation of the accessory glands. So there would be antibodies that are gluing sperms in a certain area of the semen, or in a portion of the semen. We have to first observe aggregation of spermatozoa. If the spermatozoa that are immotile and they are adhered to each other, and or there could be motile spermatozoa and non-sperm cells, they are adhered through mucous strands, there could be epithelial cells or debris, then it shows that there is an infection in accessory gland. So there is non-specific type of aggregation. It means they are haphazardly aggregated. There could be agglutination of spermatozoa that would be depicted through adherence of motile spermatozoa to each other, head to head, or tail to tail, or in mixed way. So the presence of agglutination shows the presence of anti-sperm antibodies that need further testing. And severe agglutination, that is presence of anti-sperm antibodies, would affect the assessment of sperm motility and concentration. So here we can see in this diagram the sperms, they are attached head to head. So that agglutination could be due to anti-sperm antibodies. Here it is isolated agglutination, and then less than 10 sperms are agglutinated and many are free. And then here is the moderate agglutination, here is the large agglutination, and then it would be gross agglutination. And then tail to tail could be there in less than 10, could be 10 to 50, could be more than 50, and then would be gross. So there could be tail tip to tail tip agglutination, and then it could be mixed. It could be uh, head to head, tail to tail, and they are within the same sample. Few are head to head, and then they are tail to tail. They could be tangled, mean their heads and tails, they are attached through each other in a mixed formation. So these observation could be taken on a glass slide. And this all is due to the presence of anti-sperm antibodies. It could be IgA, it could be IgG antibodies. We have further ELISA tests for the determination of these antibodies in semen. In order to determine sperm motility, we have to see slide under phase contrast microscope at 200 to 400 magnification. So phase contrast microscope, they have their application in cellular biology because they make the cellular organelles very visible as compared to light microscope. In case of light microscope, we have same background and color of the background and the stained cell and cellular organelles. But in case of phase contrast microscope, it gives brighter field to or brighter phase to the background, and it gives darker phases to the cellular organelles, so they become clearly visible on slide. So we have to assess, we have to first select 5 fields, at least 5 fields that are the visible areas under microscope for counting 200 spermatozoa, and we have to take percentages how many out of these 200 were motile, and then we would calculate how many were progressive motile, how many were total motile, how many were sluggish motile. So we'll see what are those. And then we have to observe motility after liquefaction at 30 minutes. Then what are the total motility? It is the progressive motile plus non-progressively motile or sluggish motile. They should be the reference values that we have, they should be equal to or greater than 40%, and progressive motile out of these total should be at least 32% or greater than 32%. So what is progressive motility is the spermatozoa that are moving actively, and they are proceeding in a direction, they are covering a distance. So it could be in a linear or in a large circle. And non-progressive mean they are showing all the patterns of motility, but they are not progressing, they are swimming at the same area. They are not going ahead. Say their heads are not displacing ahead. So they are just moving their flagella, but they are static in a certain area. They are trapped in a certain area. That is called as non-progressive motility. So we have to observe how many were progressively motile out of those 200 and how many were non-progressively motile. And then we could see the sperms that are entirely immotile. So in previous edition of the WHO guidelines, it was progressively motiles would be divided into rapidly progressive and slowly progressive that we said are sluggish. So rapidly progressive that move fast and slowly progressive in they are curved, they are slowly forwarding. It is again subjective term. So how we assess motility? We have to prepare the slide. We have to keep the cover slip on that, and then we have to observe it under phase contrast microscope, and we have to see 5 millimeter away from the edge of the cover slip, and so because the edges they would prevent the motility because of drying effect. So the sample it would dry at edges, and then systematically we have to scan to avoid repeating the same area. We have to change the fields randomly. So if the sample is not homogenized, it is more needed to change the fields randomly in order to be less biased. So we can go from one corner of the cover slip to another corner, and then we have to not wait for spermatozoa to swim into the field to begin scoring, because it would change our number of motile sperms or immotile sperms. And then we have to first count progressively motile spermatozoa, and then we have to count the non-progressive spermatozoa, and finally immotile spermatozoa, and then we have to calculate percentages of all these three categories. Observing sperm vitality, that is the alive and dead number of sperm, is important if the percentage of motile sperm is low. So if percentage of motile sperm is low and motile are more, so we consider they may be dead. So we have to see either they were immotile sperms were dead or alive. So we assess it by observing the integrity of the cell membrane. So if a cell membrane would intact, the we assume and it is performing its function of the differentially permeable or selectively permeable membrane, it means the cell is living. So the sperm vitality it would be measured or assessed after liquefaction at 30 minutes, and it is usually done on all samples. And then the normal values that are provided by WHO 2010, they are greater than or equal to 58% of sperms. If they are alive, it means it is normal vitality. So if high percentage of immotile and non-visible cells are present, the condition would be called as necrozoospermia, and there could be any pathology in epididymis. And if the immotile live sperms are more, it means there could be immotile cilia syndrome that we said is the uh, dyskinesia, a condition in which the flagellar protein they were not normal. There are two methods being used for determination of vitality. The first one is the most appropriate and used mostly to observe the vitality. That is dye exclusion method. So we use two dyes, eosin and nigrosin. Eosin is pinkish dye, and nigrosin is black. It is it gives background a dark color, and we can use only eosin in order to determine vitality. So in this procedure, the sperms they are stained with eosin and nigrosin. The damaged plasma membrane uh, or dead cell plasma membrane allow entry of the membrane stains through it. So the dead cell they would be stained and the alive cell they would not be stained. Then we have hypoosmotic swelling test. If live cell it is placed in a hypotonic solution, the water should enter into the alive cell. It would not enter into dead cell because membrane is so cells they would swell up if they are alive. Their membranes are intact, so they would swell up. If membranes are damaged, they would not swell. So that test is called as hypoosmotic swelling test. So, so as we said, it is one-step staining technique. Nigrosin is used to increase the contrast of the background, and the eosin that could be used alone. And we mix the semen sample and remove a 50 microliter liquid of semen, portion of semen, and mix with an equal volume of using nigrosin in a test tube, and then wait for 30 seconds, and then make a smear like we make for the blood, that is for WBC's counting. So we have make a thin film of semen on glass slide, and we then allow it to dry. So after drying, we examine it under microscope. So we observe it under light microscope at 1000 magnification, and we use oil immersion because the 1000 magnification it is oil immersion lens. And then we count the number of stained cell and unstained cell. Stained cells are actually dead cells, and unstained cells they are living cells. So we have to count 200 spermatozoa, and then we have to observe how many were living out of them. There could be disorder like leaky neck membrane. So stain is limited to the neck region, and the rest head area is unstained. It means the membrane of the neck is damaged. So there could be disintegration of cell membrane, so it means cell is alive. These cells that have leaky neck membrane, only an area of the membrane is neck is stained but head is not stained, would be these cells would be counted as alive. So here we can see these type of cells. For example, these are non-stained cells, so they are these two cells, they are alive cell. Here we can see these cells, they are dead cells. Here in this slide we can see the alive cells, they are white in color, and pinkish color is given to the dead cell. It means stain has entered into these membranes. Here in this slide we can see these cells that are white in color. They are non-stained, they are living cells, and then stained they are dead cell because eosin has entered into these cells. For vitality testing by using hypoosmotic swelling, we have to place the sperms in hypoosmotic solution, and it should the cell membrane they should swell up within 5 minutes, and there would be change in flagellar shapes, and they are stabilized till 30 minutes. So procedure of this is we have to incubate 1 ml hypoosmotic saline for 15 minutes, and we have to add 100 microliter of semen to hypoosmotic saline. Saline is sodium chloride, so it should be less than 0.9% of the concentration. Sodium chloride 0.9% is called as normal saline, and more than 0.9% is called as hypersaline, and the less than 0.9% is called as hyposmotic saline. So it should be less than 0.9%. So the water would enter into the cells, and we have to incubate it at 37 degrees centigrade for 5 to 30 minutes, and then we have to transfer 10 microliter to clean glass slide, and then we have to observe it under microscope. So we have to observe the tail. We can see the live spermatozoa, they would swell up. Here we can see they would become like this, their tails would swell up, and the dead spermatozoa, they would not swell up because their membranes are damaged, so water would enter but they would not swell up, so they would not be changed. So integration or disintegration of the membrane would be observed through the levels of swelling of the cells. The cells that are totally integrated, their membranes are working, they would be completely swell up. In this slide we can see here semen sample, and then it is now placed in hyposmotic solution, incubated for 30 minutes, and then you can see here the sperm tails, they have bent and they are being swelled, so they would be living sperms that have bent tails due to entry of water. But in case of the sperms that have linear tails, they are actually dead cells because water has not entered and trapped into these membranes. In this slide, we can also observe the sperm cells that have their bent tails, and then there are tails that are linear tails. Linear tail sperms, they are dead cells, and those that are having swelled or bent tails, it means water has entered into these membranes. For sperm numbers, as we know, we have to dilute semen because there would be more than 15 million sperm per ml of the uh, per ml of semen. That is the normal value now, and they are very concentrated. It means it is concentrated, so we have to dilute it. We dilute the semen through by using normal saline, that is 0.9% sodium chloride, and then we count it under microscope in hemocytometer. So total sperm number and sperm concentration, they are different. Sperm concentration mean number of spermatozoa per ml of the semen, or per microliter of the semen, any unit of the semen, and total sperm number mean number of sperms in total ejaculate, one ejaculate of the semen. So the normal sperm concentration would be greater than or equal to 15 million sperm per ml, and normal number of sperms in an ejaculate or total sperm number would be should be equal to 39 million or greater than that. If less than that, it means it is the oligospermia, and if it is less than sperm concentration less than 15 million, it means oligospermia, that is decrease in number of sperm cells. So we know all these steps. We have done this experiment in our class. We have demonstrated it already. We have to observe the morphology of spermatozoa. So normal morphology is that head should be consisted of nucleus with condensed chromatin, and then there would be few nuclear vacuoles in the that could be observed under microscope. And then acrosome, it would be present on the head and two-thirds of the head, and then it is 40 to 70% of the head is covered with acrosome, and it we know releases enzymes that dissolve corona radiata and zona pellucida, the cells that are present along the ovum, and zona pellucida is the membrane that should be dissolved in order to entry of sperm into the ovum. So middle piece of the sperm contains mitochondria that provides energy, and the tail is flagellum is used for the. In order to determine semen sperm morphology, we have to prepare a semen smear on glass slide. We have to take semen sample. If it is normal, then we don't have to dilute it. We have to take 10 microliter on a point on a glass slide on one corner, and then take another glass slide and making 45-degree angle, we have to drag it toward the other corner of the slide in order to make a continuous homogeneous smear. And if the sperm numbers were low that we already have determined, then we have to centrifuge this semen sample and then use the substrate and make this smear. If the semen sample has viscous material, it was viscous due to presence of certain antibodies or other material, then we have to wash it with the saline. So after washing, we have to then make the smear. And smear should be air dried, and then it would be uh, stained with PEP and short stain. We can even stain it with eosin. Eosin that we were using for the detection of the vitality of the sperms would also tell about the morphology of the sperms. And then we fix it with ethanol or methanol, and then we see it under microscope. We have to observe 200 sperms, and normal morphology would be greater than 4% of the sperms. So the head, it could be tapered, it could be pyriform, pear shaped, it could be round with no acrosome, could be small, could be amorphous, have different shapes, could be vacuolated, more than two vacuoles are there, and could be small acrosomal area that is less than 40% or could be more than 70% of the head. So there could be defects in neck and mid piece. There could be bent neck that had entered at a different than the central area of the head. It could be asymmetrical bent. It could be thick insertion into the head. It could be thin insertion. Tail defects. Tails could be short. They could be bent. They could be coiled. They could be have they could have excess cytoplasm in tails that are more than one third of the head area.